Hi everyone, my name is Günel Əyyubova. I am Senior Lecturer of Histology Department, and today we are going to start the lectures on the general histology. These will be the lectures on the cytology first of all. The topic of our lecture is the cytology as biological and medical science, the cellular theory, basic stages of formation, the modern formulation of substantive provisions, importance for biology and medicine, plasma membrane structure, functions, cortical cytoplasm and cytoskeleton elements, mechanisms of cellular movement, and also the centrosome and mitochondria. Actually, we have to know that information about the structure of the living things were started by the works of the Robert Koch, who looked and saw for the first time that in the corkscrew he saw that cork of the tree is composed of the individual subunits, which he called the cellula, which is microcompartment. And afterward, the Schleiden and Schwann they articulated the main tenets of cell theory, in which the first tenet is that all living things, all living organisms are composed of the one or more cells. Then the second one, the cell is a basic unit of structure and organizations of the organisms. So it means that the cell, it is a structural and functional unit of all life. And it is because it was discovered that it is the cell is a unit of all living things, because the cell, it has all of the characteristics that differ the living things from the non-living ones. It is the ability for the production of proteins, for the movement, reproduction, metabolism processes, energy production, and so on. So it is the cell, it has all of these abilities. Then the third tenet were articulated by afterward by other scientists. He was the Rudolf Virchow. In his work Pathology, he wrote that all of the cells come from division of pre-existing cells. So all of the new cells arise as a result of division of the previous cells. Then information about the cell structure, it also further were developed by Leeuwenhoek, who saw the living cells, which he called small animalcules. Then Robert Brown, he studied the nucleus, and so on. Then the scientists tried to study the structure of the living things, which were actual and popular for those times, and the cell theory were articulated in 1838, in 1839. Cytology, it is a science about the cell, about the structure, functions of cells, and the processes which are taking place inside of the cell. We will study in this topic and in this subject also the molecular medicine, on the molecular level. We will study all of the components of the cell. And today it is the first lecture about the general morphology of the cell, about the cell membrane. Actually, we know that all of the cells on the earth, they are of two main types. These are eukaryotes and prokaryotes. Prokaryotes, these are like bacteria. They are small, their size between 1 and 5 micrometers, and they have not any distinctive nucleus. and because they do not have nuclear envelope that separates the genetical material from the rest of the cytoplasm. But for us, it is important to know about the eukaryotic cell, because the main topic, the main goal, it is the human cells studying. That's why we afterward will talk about the eukaryotic cells, and we know that eukaryotic cells, these are cells which have nucleus, have cytoplasm, and the cell membrane. So these are three main compartments of each eukaryotic cell. And we know that the human body, it comprises more than 200 types of cells, and all of them united, and they form the tissues. Tissues form organs, organs form system organs, which is the next level in organization of the human body. Finally, all of the system organs compose whole body. That's why in this topic we are starting to study the initial, unit of the life. It is the cell. Then in further topics we will study the tissues, and afterward the organs and system organs. So we will go from the unit, from the smallest unit of life toward the more complex organism. And all of the types of cells in the human body, they arise from the one cell, which is the zygote. It is a result of fertilization of the oocyte with the spermatozoa. After the zygote divides to form new cells, these are called blastomeres. Blastomeres will form morula, morula will form blastula, then we will have gastrulation. These are steps in embryologic development, which we will study on next subject. Then we have organogenesis, during which the fetal stage or embryonic stage will create the human body, and during embryonic stage all of the system organs will be established. Then further growth and differentiation will create this body, the body of the adult human. What we have to know from here, that the zygote and the first blastomeres that formed as a result of division of the zygote, all of them, they are told to be totipotential cells according to the potentiality to form the new cell types. And totipotential, it means that these cells are totipotential. It means that they may give rise to so much, all of the cell types of the body, but also to the extra embryonic organs like placenta, then will be the yolk sac, the amniotic cavity, chorion, and others. All of them will be able to develop from the zygote, so it is totipotential cell, and also blastomeres. If we will separate these blastomeres, each of them have ability, or totipotential ability. Then from these cells will be formed the blastula. Blastula has outer cell mass, inner cell mass, which is called embryoblast. So from the embryoblast cells will form the embryo proper, and from the trophoblast cells will develop the extra embryonic organs. It means that embryoblast cells, they may form just human body and not also the extra embryonic organs. That's why these cells decreased their potentiality, and in this case they are told to be pluripotential cells. So pluripotential cells, they are more committed, more determined than the totipotential cells. So these inner cell mass cells will form the germ layers, ectoderm, mesoderm, and endoderm germ layers, from which will develop certain system organs. And then on next level we will have gastrulation. There will be formed epiblast, hypoblast, germ layers, and from them will develop other. So the potentiality of cells with each step of differentiation will decrease, and the cells which come afterward, they will become multipotential, oligopotential, and finally monopotential or unipotential. It means that from unipotential cells will develop just one cell type and not just one cell type. It's more committed, determined to become certain cell type just. General morphology of the cell. Each cell has the membrane, just we told about, cytoplasm, nucleus. We have in our body cells which have many nuclei, multinucleated cells also. They are formed by two ways. It's first way when the nucleus subdivides, so karyokinesis we will have, division of the nucleus, which will not be followed by division of the cytoplasm by the cytokinesis. So the multinucleated cells will be created, for example, in liver cells, which may have two or more nuclei. In second way, of the formation of the multinucleated cells, it's for example in the muscle fibers, myosynplasts or syncytiotrophoblasts. They are formed as a result of fusion of different various amount of cells. They fuse with each other and they create large sacs that reach in nuclei. And that will form the syncytia and symplasts, so multinucleated cells. We have in our body also cells which do not have nucleus, for example, erythrocytes, red blood cells, and corneocytes of the stratum corneum of the epidermis, which during differentiation lost the nuclei. So the cytoplasm of the cell. Cytoplasm, it is subdivided into two components, that is the cytosol or cytoplasmic matrix, which contain organic and inorganic substances that may be dissolved or may be present in the state of the gel. And inside of the cytosol we also have organelles which do not have membranes, and also cytosol includes cytoskeleton elements. These are filaments. Then here we are looking at the microtubules. These are cytoskeleton elements. And non-membranous organelles like centrosome, which contains centrioles. And also we have ribosomes and proteasomes. They form cytosol. And also in cytoplasm we have vacuoloplasm, which mean membrane-bounded compartment inside of the cytoplasm. It means that this is actually our membranous organelles. They are rough endoplasmic reticulum, Golgi apparatus, lysosomes, peroxisomes, endosomes, and mitochondria. They are all organelles which have membranes, and together all of them will form microcompartment, membrane-bounded microcompartment inside of the cytoplasm, and that is called vacuoloplasm. Then on the surface of the cell we have microvilli. These are finger-like projections, that extensions of the cell which increase surface area of the membrane, which is needed for different purposes. Maybe movement, maybe absorption of different various substances, and so on. In electromicrograph representation of the cell, we can see all of the mentioned components: the nucleus, cytoplasm, membrane of the cell, and here we are looking also at the organelles and secretory vesicles. These are large, numerous rounded structures. These are secretory vesicles. These are product secret of the cell, which is stored inside of these vacuoles, and when it is needed they are released into the outside of the cell. Here we can see the organelles. just we told, and that is a classification of the organelles, subdivision of them into membranous and non-membranous. The cytoplasmic matrix, just I mentioned that this is a concentrated aqueous gel containing molecules of different sizes and shapes. The electrolytes here, the RNA, synthesized proteins, and it is the cytophysiological processes that are fundamental for the living of the cell. For example, protein synthesis, breakdown of nutrients, that is the cytosol. And now we are coming to the first compartment. It is the cell membrane. And the cell membrane, it covers the cell. It is a barrier between the inside and outside environment of the cell. Here we have to mention about the theories which were developed at the investigation of the cell membrane structural composition. It was made on the example of the erythrocytes, red blood cells. And first of all, the Görter and Grengel, they proposed double lipid model, according to which the cell membrane, it includes two layers, outer layer and inner layer of phospholipids. So phospholipids form the layers, complete layers. Each of the phospholipid molecule has hydrophilic head, which have green color here, and hydrophobic tail that include two chains. One of them straight, other one is curved. And these chains, they are hydrophobic, they composed of the fatty acids. And these hydrophobic tails, they face each other, and by that way there is formed barrier that isolates inside from the outside of the cell. Then it was announced, it was become clear that the cell membrane, it includes not just lipids, but also the proteins. They form complete layer outside of the membrane and inside. And that is sandwich theory, which were proposed by Daniele and Dawson. They thought that the membrane include also proteins, which are they form coats on the surface of the phospholipids. But afterward, by applying cryofractography methods for studying of the cell membrane, in which the cells are freezing at minus 196 grade, then the cells, then the membranes are cleaved, and this breaking, this cut were made between phospholipid layers, and the inside of the membrane will be open, and their scientists saw that the proteins, they do not form complete layers, but they are scattered through the membrane as a mosaic. Here we can distinguish integral proteins, which span all of the thickness of the membrane, and this one also, and peripheral proteins, which attach to just one layer of phospholipids. These integral proteins, they are amphipathic, in which they have amino acids that are hydrophobic in nature and in their characteristics. They are attached, they join the phospholipid molecules, and they have hydrophilic amino acids, which located in the center of these proteins. Some of the integral proteins are multipass proteins, in which their alpha chains pass through the membrane several times, and they are longer. That's why they have specific sites for binding with various chemical substances outside and the inside of the membrane. That's why they called transmembrane proteins. And this theory, which is accepted, that is called mosaic fluid model, in which mosaic, it's according to the location of the proteins, and fluid, it means that cell membranes is jelly state substance. It is not hard, it's not gas, it's not but it is the gel state fluid. This ability is imparted by the phospholipid molecules. Here we are looking at the phospholipid bilayer, and in which proteins are embedded, some of them attached to just one layer. And outside of the membrane, we are looking at the carbohydrate chains, which are located outside of the membrane, and some of them may join the lipid molecules, in which this molecule will named the glycolipids, or to the proteins. In this case, they will call glycoprotein molecules. And so carbohydrates of the membrane, they form complete layer coat around the cell membrane. This coat is called glycocalyx, which protect the cell from the chemical, mechanical, and physical injury. Also, glycocalyx participate in recognition in the receptor function of the cell membrane, because the glycoproteins, each of them, they serve as a receptor for recognition of certain molecules. And also glycocalyx, here we have to mention some structures which we can see here. They called lipid rafts. Inside of the lipid rafts, we can distinguish different integral proteins, also phospholipid molecules. There are sphingolipids. Actually, phospholipid molecules are also of different types. Sphingolipids, or we have inositol phosphates, and also inositol, and also some other phospholipid molecules because of their chemical composition. In this case, lipid rafts contain also cholesterol molecules. So these form complexes that they are functional complexes which are needed for very fast transmission of information, of the signals from the outside to the inside of the cell. They are like floating signaling platforms, because in this inside of these platforms there are all of the components which are needed to receive signal and to transmit it to intracellular events. Besides it, inside of the membrane, we have cholesterol molecules. And cholesterol molecules, we can see them here. They are located between the phospholipids. Their hydrophilic OH groups attach to the hydrophilic heads of the phospholipid molecules, but their carbohydrogen chains attach to the tails, hydrophobic tails of the phospholipid molecules. And besides, we have to mention that lipids, cholesterol, phospholipids, together with some of the proteins, they may join each other and form lipid rafts. The rafts, they are complexes of lipids and proteins, which serve together, they function together, and they provide fast passage of signals into the cell. On the surface of the membrane, we have carbohydrates, the green chains here. The carbohydrates may attach to the proteins. In this case, they form glycoproteins, or they may attach to the lipids of the membrane. In that case, they will call glycolipids. So, carbohydrates of the membrane, they are present inside of the glycoprotein molecules or glycolipid molecules, and they located outside of the membrane. Here they form one layer outside of the membrane. It is called glycocalyx. The glycocalyx protect the cell from the physical, chemical, or mechanical injury. Also, carbohydrates they serve as receptors here, and they provide receptor function of the cell membrane. Glycocalyx is stained because it rich in carbohydrates. It's stained with Alcyon blue and other specific stains. Then that is the electron microscope representation of the cell membrane, and under the electron microscope we can see here two cells. That is one cell, endothelial cell, that line blood vessels, and this is the other cell. So two neighbor cells we are looking here. That is the membrane of one cell, and that is the membrane of the other neighbor cell. And here they come together, they come very close to each other. Under the electron microscope, we can see that membrane includes three layers, two electron dense layers, between them one electron lucent layer. So it has three layered structure under the electron microscope. The diameter of the membrane is 8 nanometers. The dark regions and the light region of the membrane, it is because of accumulation of osmium salts at the hydrophobic parts of the membrane. That's why it will have the three-layered composition under the electron microscope. Here we can see the fluid mosaic model of the cell membrane, and these are the phospholipid molecules which form double layer, cholesterol molecules, then glycoproteins, glycolipid molecules, peripheral proteins, outer and inner peripheral proteins of the cell membrane. If phospholipid molecules, they increase fluidity of the cell membrane, increase permeability of the membrane, but cholesterol molecules decrease that capabilities. Phospholipid molecules include hydrophilic head, which contain phosphate group, glycerol, and other additional group. And there are two chains, each of which composed of the fatty acids, but one of them is composed of saturated fatty acids, other one composed of unsaturated fatty acid chains. By that way, the other one provide movability to the straight chain. So one of them is curved, other one is straight. Because of this bending, the straight chain have ability for movement, and it will provide fluidity of the membrane. The cholesterol molecule here, it linked to carbohydrates or phospholipids and limit a movement of plasma membrane. So they reduce a fluidity and deformation of the cell membrane. Cell membrane has some functions. They are, the cell membrane, it is the coat that covers the cell, and it's not just only barrier. It provides exchange of substances. It provides structural support for the cell. It provides interactions of the cell with the outer, with cellular and non-cellular components that surround the cell. That's why all of these functions we can join in 5 functions. First of all, it is the selective permeability, then endocytosis, exocytosis, receptor function, and intercellular interactions. The first function of the cell membrane, it is the selective permeability. We have to know that selective permeability of the membrane permit entrance and exit of just the substances which are needed for the cell. So it's very selective filter the cell membrane. We know here that through the membrane there is taking place the passage of substances which can be dissolved within the lipids. For example, in this case we can see the oxygen, carbon dioxide, these gases, and then here we are looking at the hydrophobic substances like benzene, Then the hydrophilic or small polar molecules like water, ethanol, they may pass through the cell membrane because they can be dissolved within the lipid bilayer of the membrane. But large polar molecules like glucose, amino acids, they can't pass through the membrane, as well as the ions like sodium chloride and other charged molecules. They also cannot pass through the membrane easily. That's why for their passage we have two kinds of transportation through the membrane. Here we can see the passive transport and active transport. Passive transport have two modifications, that is a simple diffusion and facilitated diffusion. Both of these kinds of transportations, they do not need any energy, and they are taking place down the concentration gradient of substances. It means that substances will be passed through the membrane from the side of the high concentration to the side of the low concentration. If this process is taking place without aid of anything, we mentioned hydrophobic, small polar molecules, and the gases. They can pass through the membrane via simple diffusion. The facilitated diffusion provides passage of substances also down concentration gradient from high to low, but there is needed some aid. This aid is the integral proteins of the membrane, which may form here ion channels or the carrier proteins, and both of them will provide facilitated diffusion without aid of energy and by the integral proteins. These proteins said to be amphipathic because they have hydrophilic amino acids which form core inside of the protein, and hydrophobic amino acids which face the lipids of the membrane. Then we have active transportation. The active transport provides by expending energy. The cell can transport substances and small molecules, ions against the concentration gradient, as in this case the substances from the side of their low concentration, they will be carried to the side of their high concentration, but that process needs energy, that is the energy of ATP. So we are coming to the channels. We have ion channels that are formed by channel proteins, and the channel proteins may be gated or ungated. And the gated channels, these are channels for sodium chloride, then potassium ions, and we have ungated or open ion channels. They do not have any gate. They are channels for the water. Then we have, for example, the water ungated channel, and through this channel they called aquaporins. They are ungated. They have shape of watch glass. They're formed by multipass proteins, integral proteins of the membrane. In the center of this channel we have some filter, and this is the filter is formed by 4 amino acid subunits, and the narrow sides and the electrostatic pushing of positive charged ions, the amino acids, they will ensure the turning pass of water molecules from the channel, and water molecules will turn, and in the center, because the both sides of the channel is surrounded by positively charged amino acids, they will repel the water molecules, and it will lead to turning of the water molecules in the center of the channel, and that's why they will pass the water molecules. There are 13 types of aquaporins in the membrane. They are signed by aquaporin 0 till aquaporin 12. Depending on their location, they are in various modifications. And also, let's look at the gated ion channels. The gated ion channels, it means that they have some gate, and for example, in this case, that is a potassium channel. It is formed by 4 polypeptide chains, 2 in this side and 2 in this side. This is an illustration which were performed by McKinnon, and McKinnon, he was the scientist who were awarded by Nobel Prize because of the discovery of this channel, and this is the illustration which were taken by him, prepared by him. And here in the center of this channel we have ion filter through which the potassium ions will pass. But before passing the potassium channels, they inside or outside of the cell, they are bound to water molecules, and when they come to the filter, here we are looking at the filter, selective filter of the channel, the potassium ions lose their water and they pass through the center of the filter, and this filter is surrounded by carbonyl groups, CO groups, by oxygens of these carbonyl groups. And here the potassium ions will form electrostatic bond with those oxygens. By that way it will pass through the filter. After passage, the potassium again will bind water, and how it will pass then. After crossing the filter, the ion channel need to be open. And here we can see the gate, which is closed actually at the rest state. So the channel needs some signal which will cause opening. Here we can see the closed state, and in this case it is the open state of the channel. So the channel needs some signal to be open. That is a sodium ion channel. It's also a gated channel. By the way, it has some activation gate and inactivation gate. At the rest, activation gate is closed. Sodium channels are actually voltage gated channels, most of them. It means that the ion channels, gated ion channels, they are classified according to the mechanism which will cause the opening. They are voltage gated, they will open under the influence of wave of depolarization. And ligand gated channels, they open under the influence of the neurotransmitter. Ligands, we know that they are various chemical substances. They may be neurotransmitters, nucleotides, or G proteins. So these are the substances which, after binding to the ion channels, they will cause their opening. And so we have also mechanically gated channels. They will open under the influence of some mechanical stimuli, maybe deformation of the cell. And so the sodium channel, it is voltage gated. It will open under the influence of wave of depolarization, which will reach the channel, and it will cause opening of it. Now, after opening the activation gate, in response to a nerve impulse, the gate opens and sodium enters the cell. 1 millisecond after opening, this channel will be closed by inactivation gate, and this state of the channel is called refractor state. It is inactivated for a brief period following activation. The channel does not open in response to a new signal, that is a refractor state. Then it will be followed by this state. It is so finally this activation gate will be closed. That's how sodium channels operate. Sodium channels, they most of them, they are responsible for depolarization of the cell. When the sodium ions enter the cell, the amount of the sodium ions inside of the cytoplasm increase, and it leads to depolarization. inside of the cell. Actually, we know that at the rest state, our cells have minus 90, minus 70 millivolt charge, and entrance of positively charged ions into the cytoplasm will cause changing of that resting potential to the in positive direction. So it will lead to excitation of the cell. And here the carrier proteins, they are also integral proteins, but their operation is different in which the carrier proteins have specialized sites for binding with the ligand with that ion to be transported, and then they change their conformation, and by that way the substance will be transported to the opposite side of the membrane. So they are also multipass transport proteins, and that is how the carrier proteins will work, but also participate in facilitated diffusion. It's also a type of passive transport. It means that they also do not need energy. Here we can see the carrier transportation by carrier proteins. Here we have the carrier proteins which carry just one substance. This carrier protein is designed to carry just amino acids. We have also the carrier proteins which carry just glucose, and these are the symport transport, antiport transport. Symport, in which two substances will be carried by one carrier protein in one direction, in the same direction. In antiport, they will be transported in opposite directions. That is how uniport transporters. We have symport, antiport. These two belong to coupled transport. And the active transport. In active transport will participate ATPases or pumps that are of different types inside of our cell membranes. They are classified according to different features. In here we can see P-type, F, V-type, ABC type of ATPases. They participate in primary active transport, and secondary active transport. We also have this one here. Let's look. Passive transport, simple diffusion, ion channel gated transport, carrier protein gated transport. All of them carry substances from high concentration to low, so that is passive, that not need energy. And the active transport, we have primary, when one substance goes in one direction, but it is the main thing here, that is from low to high concentration. And in this case, secondary active transport will be possible because of the energy of the primary active transport. Here the substances go in from one side of the membrane to opposite. Here we can see them, for example, yellow balls from high to low concentration come, but green balls from low to high, because they will be carried by the ions which went out because of the primary active transport. That will be secondary active transport. Here we can see sodium potassium pump operates. It's one of type of the ATPases or pumps that carry 3 sodiums outside of the cell and 2 potassiums inside of the cell. Then that sodium ions will increase in amount in the outside of the cell. In extracellular fluid we have increased sodium ions, and then sodium ions will return to the cell cytoplasm, and they will come down the concentration gradient from high to low concentration, but sodium ions will carry the other substance, that is glucose, for example, that will be carried against concentration gradient. Here we can see sodium and glucose. They are transported in one direction, in the same direction, but sodium down concentration gradient, but glucose against concentration gradients. That is the secondary active transport. Here we can see how operates sodium potassium pump. 3 sodiums bind to pump. Then here we can see a phosphorylation of the pump, or this glycoprotein part of the pump, or ATPase. Phosphate binds to this pump, and as a result, this pump will change its conformation and 3 sodiums go out. Then 2 potassiums from outside of the cell bind to this carrier protein or to the pump, and as a result, the pump here it undergoes dephosphorylation. Phosphate detaches from the glycoprotein from this carrier, and finally this carrier will return to its previous conformation and 2 potassiums enter the cell. That is how the pump operates. And endocytosis, another function of the cell membrane, it's uptake of large amount of fluid or particular substance from the outside, and take up and ingest this substance in the formation of the vesicles. There are 2 types of endocytosis. It's phagocytosis and pinocytosis. Phagocytosis, it's ingestion of particular matter by the cell, and pinocytosis, it's ingestion of the large amount of the fluid and the substances which are dissolved within the fluid. Pinocytosis, there are 2 kinds of it. Macropinocytosis, the vesicles have large diameter, 250 nanometer approximately, and here we can see the micropinocytosis, the caveolosomes, simple pinocytosis without any coat. Here the caveolin protein, and that is a receptor mediated endocytosis. Then in receptor mediated endocytosis participate some components. There are more than 20 types of proteins which participate in this process, but what we have to take in mind that the ligands which are carried to the cell, they have receptors on the cell membrane, and after binding ligands to the receptors, there are other proteins, adapter proteins, which bind AP2, bind to another end of receptor, and the clathrin proteins will also accumulate here, and they change their conformation, and the cell will form pit that after the beat they becomes deeper, and finally there are vesicles formed. At the neck of the vesicle accumulate dynamine and epiphysin proteins. That will dynamine have GTPase activity, and it will lead to detachment of the vesicle from the membrane. Then the vesicle will lose its clathrin coat, and the endocytotic vesicle enters the cytoplasm. Exocytosis, it's a release of the process by which the vesicle moves from the cytoplasm to the cell membrane, where it discharges its content outside into the extracellular space. There are 2 general pathways for exocytosis. It's constitutive pathway and regulatory. Here participates snare protein, snare receptor, snare T snare, it's located as a cell membrane, and V snare or vesicle snare or synaptobrevin, the green one is synaptobrevin, the blue one is T snare or syntaxin. Between them will be formed bridge by the proteins SNAP25. They will bind each other, and vesicle will be moved toward the cell membrane and fuse with the membrane to discharge its content outside. The energy required process, this one is 1 ATP here will be broken, and other ATP to return all of these substances, all of these components to their previous conformation. Receptor function of the cell membrane. The cell has receptors. For each ligand, the cell membrane has specific receptors like key and keyhole. So in our body, in our multicellular body, the cells interact with each other, in which one cell produces some substance, the chemical substance. It is a ligand. It binds to the receptors on the target cell. And according to types of signaling cell and target cells, there are different types of signaling mechanisms in the human body. For example, intracrine signaling, when ligand or chemical substance which is produced by the cell will influence the cell itself and without going outside. That is autocrine. When the cell produces ligand, it goes outside of the cell, then binds to the receptors on the surface of the membrane. Then we have juxtacrine signaling, when the signaling cell and target cell, they are neighbor cells. One of them produces ligand, other one binds via its receptors. Then here we are looking at the paracrine signaling, when the signaling and target cells locate at some distance. Then matricrine and endocrine mechanism, when the ligand is released into the blood circulation, and together with blood it will be carried to the target cells. Neurotransmitter influence, that is synaptic connections, that is neurosecretory cells that produce hormones into the bloodstream, into the blood circulation. So neurohumoral signaling and neurotransmitter signalings we also have. Second messengers, what are they? So if we have signaling molecule, that is first messenger, and we have receptor which bind that ligand, then after binding of the ligands to the receptors, in the cell will take place activation of the system of second messengers. The second messengers are needed to carry the message from the signaling molecule to the intracellular events, to the activation of some processes inside of the cell. It may be production of new proteins, it may be increase of the amount of some ions, and so on. For example, let's look here, G protein. G protein is also second messenger, which at the rest binds GTP. When the receptor is activated by the ligand, G protein is activated. It leads to activation of phospholipase C enzyme, and it will break from the phospholipids of the membrane deacylglycerol or the inositol 3-phosphate. Both of them, these are second messengers. Then these second messengers, for example, inositol 3-phosphate comes and binds to its receptor on the smooth endoplasmic reticulum and activates here the calcium channel, and the calcium channel after activation opens, and the calcium ions go outside to the cell cytoplasm. The amount will increase and it will lead to some cellular response. So we have different cell surface receptors like ion channel linked receptors, 7-spiral and enzyme linked receptors, cell adhesion receptors, and nuclear receptors. All of them, they are receptors which we have in our cell. Something I would like to talk you about the receptors which located in the cell membrane. So these are all of them, these belong to the cell membrane. They are located in cell membrane. The 7-spiral, this one, enzyme linked receptors, these two, and cell adhesion receptors, integrins, caterins, and selectins. And we have also ion channel linked receptors. They all belong to the hydrophilic substances which cannot pass through the cell membrane. That's why they bind to the membrane receptors. But hydrophobic substances, they are receptors located inside of the cell, and they called nuclear receptors. So these are kinds of second messengers, which we have G protein, cyclic nucleotides, CAMP, cyclic GMP. We also have phospholipid derivatives, inositol 3-phosphate, just I mentioned, the diacylglycerol, ceramide, also nitric oxide. Okay, let's come to the cytoskeleton. Cytoskeleton, this is a skeleton of the cell which provides its shape, and there are three components which make up the cytoskeleton. These are thin filaments, microfilaments, intermediate filaments, and microtubules. All of them, these are microtubules here, microfilaments which have small diameter. And here we also can see something about the cortical cytoskeleton. In our cell, we have skeleton which is inside of it, and we have cortical cytoskeleton which is located under the cell membrane. It is cortical cytoskeleton which make up the cortex of the cell, which provide its shape. It provides a passage of signals into the cell. For example, in the case of the erythrocytes, red blood cells, we are looking at the cytoskeleton elements. The cortical cytoskeleton core is formed by the spectrin protein, which form here helix, double helix, and it attached to the cell membrane via specific additional proteins, for example 4.1 protein, ankyrin, pallidin, and anion exchanger. We have here the red large one. Other glycoproteins, the glycophorin is bind to the 4.1 proteins, and all of this configuration will be needed to maintain the shape of the cell, to maintain its elasticity. And here we have the actin protein. The actins form double helix. It has positive end which bound to the adduction, negative end binds to the tropomodulin, and so they provide shape of the cell. Actin filaments, these are about the endoskeleton or internal skeleton. First of its components, this is the actin filaments. They have role in pseudopod formation, in endocytosis, exocytosis, in movement of organelles inside of the cytoplasm, and localized in microvilli also. And they form junctions in epithelial cells. They are needed for mitosis and cytokinesis of the cell when the cell subdivides into two parts. So here we are looking at the distribution of actin filaments in human fibroblast in culture, and that is under the electron microscope. Here the scanning electron microscope. This is the actin filaments which form terminal web under the cell membrane. Actin filaments, this one, they have a diameter approximately 6 nanometers. Microfilaments, they formed by the protein actins, which are globular actins that bind to each other and form fibrillar actins. Two chains coil around each other to form double helix. Each of them have plus end and negative minus end. Intermediate filaments, they are formed by the 8 tetrameres, which form intermediate filament with diameter 8 to 10 nanometers. And finally, microtubules, which formed by tubulin proteins. There are alpha and beta tubulins that bind to each other. Then they form protofilaments, and 13 protofilaments will form one microtubule, which is like a cylinder. It has cylindrical shape. It also has plus and negative minus end. Microtubules make up the centrioles. This is the organelles. This is the organelle of the cell, which that form centrosome. Two centrioles form one centrosome. And inside of the centrioles, we also can find microtubules. There are 9 triplets of microtubules which form one centriole. So actin filaments form a dynamic network important for cell shape. change such as those during cell division, locomotion, and formation of cellular processes, folds, pseudopodia, lamellopodia, microvilli, etc., which serve to change a cell's surface area to give direction to a cell's crawling movements. Acting as a microfilament in cell, we are looking here. And intermediate filaments. We have to know that intermediate filaments in different tissue types, they made up of different proteins, and that is a distinguishing feature, and this feature is used in histochemical diagnostics for the sources of tissues from which develops any pathological formation. For example, if we have to make diagnostics of tumors, we use histochemical methods to evaluate the type of intermediate filaments inside of it. By that way, we can find the source of development of such a tumor. For example, cytokeratin epithelial, cytokeratin here, it is the intermediate filament protein for epithelial cells. Vimentin in mesenchymal cells, desmin in muscle cells, glial fibrillar acid protein in astrocytes in the glial cells of the central nervous system. The neurofilaments we can find in neurons or nerve cells, and finally nuclear lamins in all cells in the cell inside of the nucleus under the nuclear envelope. They are lamine proteins. And then the average diameter of the intermediate filaments is from 8 to 10 nanometers. It's between actin filaments and microtubules. And they serve to provide mechanical strength and stability of the cell. So intermediate filaments do not participate in the movement of cells. And here we can see skin epithelial cells showing intermediate filaments of keratin associated with desmosomes. So these are the regions of desmosomes, intercellular junctions, and inside of the desmosomes we can find these intermediate filaments. And here we also can find intermediate filaments in epithelial cells. And nuclear lamina is formed by lamine proteins, which also that type of intermediate filaments. Here we can see the classification of these intermediate filaments, the keratins, then here desmin in muscle, vimentin in mesenchymal tissues, glial fibrillar acid in the glial cell, neurofilaments in neurons, and finally nuclear lamins in the cell nuclear lamina. That is also you can see the electron microscope representations of actin filaments, intermediate filaments, and microtubules. This number 3 here. Microfilaments, intermediate filaments, and microtubules. This illustration you have in your atlases. Here we are looking at the microtubules in cells. That is a confocal microscopic appearance of the microtubule network inside of the cell. And in this case we can see the microtubules which form the mitotic spindle apparatus during cell division. And here we have to pay attention that all of the microtubules arise from the centrosome. Centrosomes, these are organelles that include periocentric matrix, and that here located microtubule organizing center. So from the microtubule organizing center radiate microtubules in all directions, and also between poles of cells or between poles of one cell will be formed these microtubules. Then they will assist in division of the cell. And here we also can see there one of the microtubule structure. Actually, in electron micrograph representation, we also can see these microtubules. These are like cylinders in fibroblast cytoplasm. And so they composed of tubulin subunits, we have to remember, and they are oriented in arrays. Then they generally extend from the centrosome area near the nucleus. They extend from the centrosome area, and they are arranged towards the peripheral part of the cell cytoplasm, and they serve to stabilize the cell shape. They form also the tracks for now we will look at tracks, but here pay attention to the microtubules and also actin filaments. Microtubules have green color, but actin filaments have red color, which locate under the membrane. They form a terminal web under the cell membrane, actin filaments. It's in cultured cell. And yes, here we can see that the centrosome in the center, and from it we have tracks, railroad tracks that resemble these microtubules. They are minus end attached to this centrosome, and they are plus end directed toward the cell membrane. And on the surface of these tracks move these motor proteins. They are dynein and kinesin. They are motor proteins. And pay attention, kinesin, it's transport of vesicles and organelles into the cell periphery. And here is written also the dynein, which provide transport, that is dynein, which provide transport towards the center. One end of these proteins, motor proteins, attach this organelle or the whatever inside of the cell, or maybe vesicle, secretory vesicle, and by that way they will move step by step on the surface of these tracks, and they will carry these organelles, these structures inside of the cell. They will provide intracellular movement of cellular components from one side of the cell to the other side. These motor proteins also called chemomechanical exchangers because they use energy of ATP, break that ATP, release energy, and use that energy for any mechanical action, in this case the movement. That's why they called chemomechanical exchangers. And we have to mention also agents that prevent microtubular function. Just we told that microtubules composed of the alpha and beta tubulins, each of which include minus and plus charges, and they join each other to form protofilaments. They are elongated structures, and protofilaments join side by side to form the microtubule structure. Here we'll have to remember that colchicine, it is the drug that binds to tubulin dimers and prevents the assembly into the microtubules. And vinblastine, and vincristine, they used as anti-tumor therapy. Also inhibit tubulin polymerization. So vinblastine, vincristine also prevent the polymerization of the tubulins to form microtubules. All of them they used for as anti-tumor therapy to kill tumor cells. Also, we can use nicodazole. This is another protein inhibitor for tubulin polymerization. And here we can see tubulin colchicine complex, and that it will prevent polymerization. There is another drug that is the Taxol. It binds to microtubules, preventing the depolymerization. Taxol disrupts mitosis by affecting the dynamic assembly and disassembly of the mitotic spindle required for separation of chromosomes into daughter cells. So antimitotic drugs are potent inhibitors of the polymerization and depolymerization of the microtubules of the mitotic spindle. And antimitotic drugs, they bind to diverse sites on tubulin, and the combination can be therapeutically more efficient. Here we can see the microtubules and basal feet of centrioles. By that way, by preventing polymerization of the microtubules, they prevent also mitosis, mitotic division. It's very important in case of the tumor cells. And here we can see the centriole. Actually, our cytoskeleton elements, they form different types of projections. The microvilli, maybe also cilia. In this case, we can see the cilia structure. They are hair-like projections on the surface of the cells, and from each cilia starts from the basal body. At the base of the cilia we have basal body, which has the structure same as in the case of the centrioles. There are 9 triplets of microtubules. They signed as A, B, C, and from this basal body starts axonema, that is a core skeleton for the cilia. This cilia, it includes 9 doublets of microtubules, as we are looking here, 9 doublets of microtubules and 1 pair centrally located microtubules. Around the centrally located microtubules we have central sheath. From the central sheath we have projections. They are spokes that radiate from the center toward these doublets of microtubules. Microtubules, they signed as A and B, from which A has 13 protofilaments, it has complete structure, but B, it has incomplete structure, it has 10 to 11 microtubule protofilaments, because they share 2 or 3 of the microtubules of the A, protofilaments of the A microtubule. And between the microtubule doublets we have nexin. It is the nexin, this is the elastic protein, and besides it we have dynein arms, 2 arms between these doublets. And this dynein, it as we know, that is motor protein, and they will provide sliding movements of microtubules, and they will provide movement of the cilia. Cilia, they are present on the epithelial cells that line our respiratory system, beginning from the nasal cavity till bronchioles. And by beating, they move mucus on the surface of epithelium, and the dust particles and foreign things which are entrapped within the mucus, they will move due to beating action of cilia towards the oropharynx, from where it will be expectorated or swallowed. By that way, respiratory system will clean itself. Okay, now we are coming to the cell movement. Actually, in the body we have 2 kinds of cell movement. First of all, that is slow movement. Second one, fast movement. Slow movement, it's we can demonstrate in the case of fibroblasts, connective tissue cells, or endothelial cells that line the blood vessels internally. And so in this case, in slow movement, the cells adhere to the surface on which they will move. There are adhesion molecules which provide attachment of this moving cell to the surface. Then on the anterior pool there is formed filopodia, that is finger-like extension, and inside of the cell cytoplasm Because of the assembly of actin filaments together with myosins, there are formed contractile bundles, and by activity of the contractile bundles, the content of the cytoplasm will move towards the filopodia. Then the posterior pole will be dragged towards the middle part of the cell, and finally by that way the cell will move forward. But in second type of cell movement, we can see the attachment adhesion of the just middle part of the cell to the surface on which they are going to move. That is in case of T lymphocytes and neutrophils. These are white blood cells that arise at the place of inflammation. In this case, in the anterior pole, there is also formed some extension, but that is a lamellipodia, which is more wider than the filopodia. That's why the content, intracellular content of the cell, which by the activity of the contractile bundles formed by actin and myosin filaments, they will more fast towards the anterior pole. Then the posterior pole will be dragged towards the center, and by that activity the cell will move. Here we have to mention about specialized adhesion molecules. It's intercellular junctional molecules, the lymphocyte factor A. They are adhesion molecules that will aid in cell movement. So that is about the cell movement, and that's all for today's lecture. Dear students, see you on next lecture.